Hello and welcome to my next video on hormones. Start off with some definitions. Hormones are molecules that act as messengers carrying a signal from an endocrine gland to a specific target organ or tissue. An endocrine gland is a gland that secretes hormones directly into the blood. It does not contain ducts. And an exocrine gland is a gland that secretes molecules into a duct that carries the molecule to where they are used. Hormones are secreted when an endocrine gland is stimulated. So hormones only come out of endocrine glands. These glands can be stimulated by a change in concentration of specific substances or stimulated by electrical impulses. And then these are they go directly into the blood, where the blood carries around all the body, and all the cells have the chance to be targeted by these hormones. Now, these cells, target tissues, will have specific complementary receptors which the hormone will bind to and stimulate a response. If a cell does not have a receptor on it, the hormone will ignore it. There are two main types of hormones. There are protein hormones, which are not soluble and do not enter the cell. They just bind to the cell membrane. And steroid hormones, which enter the cell and have an effect on DNA. Steroid hormones can do this because they are based on cholesterol. Cholesterol is found in the phospholipid bilayer, and the phospholipid bilayer is permeable to cholesterol, so it can go through and have effect on DNA. One example you need to know about is adrenaline. And this is a process that works very similarly to a lot of different hormones. Firstly, adrenaline is a protein-based hormone, so that means it'll bind to the cell membrane and not enter the cell. So it'll bind to a receptor site, and what adrenaline will do is it'll cause an enzyme called adenyl cyclase to be activated. Now, adrenaline is called the first messenger. It causes this response, and then there's a second messenger which causes the actual response inside the cell. So, adrenaline binds to the site and activates adenyl cyclase. Now, what this causes, since it is a catalyst, will cause the production of the second messenger, which is CAMP or cyclic AMP. Now, this activates what we call a cascade. So enzyme reactions, more glucose available to the cell, and this is what causes the reaction inside the cell. And the organ that releases adrenaline is the adrenal medulla. Now, a medulla is just a section of something you'll have a look at later as part of medulla in the brain and in the liver, but here the adrenal medulla releases adrenaline. The adrenal glands where adrenaline is created, they are endocrine glands because they're releasing hormones and are found just above the kidneys. The pancreas. The pancreas is an endocrine and an exocrine gland. So it has a function of both. It is found just below your kind of stomach. It's a leaf-shaped organ. So most of the pancreas is exocrine tissue. These are acinar cells or acinar cells. They're found in clusters around the pancreatic duct, because remember, exocrine glands have ducts. Now they're found in clusters, and they will secrete digestive enzymes into the pancreatic duct. The enzymes digest food in the duodenum, and so they have lots of different enzymes. They have amylase, which breaks down starch. You have protease, which breaks down proteins, and lipase, which breaks down lipids. And this is all in there. There's also sodium hydrogen carbonate. This is all in the pancreatic juice, as we call it. The endocrine function is much more important because this controls blood glucose concentration. There's something called the islets of Langerhans. Now, these are found in clusters around blood capillaries because, as an endocrine, they are released directly into the blood. Now, the islets of Langerhans secrete hormones, and there are two in particular: glucagon and insulin. And in the islets of Langerhans, there are two types of cells: alpha cells, which secrete glucagon. Remember that because alpha glucagon, it has an A in it, glucagon, and beta cells secrete insulin. Now, the regulation of blood glucose concentration is a negative feedback control. So when you eat, you will get a lot of sugar going into your body, and it will raise the level of blood glucose in the blood to a higher level than normal. This will be detected. But also, if you break down a lot of it, you will then have a lower blood glucose concentration, so that will also be detected. Now this is detected in the islets of Langerhans. If there is a rise in blood glucose concentration, the beta cells detect it and release insulin. This goes to liver cells and muscle cells. Now, an important thing to say, which is very important in particularly A2, the beta cells start producing insulin, but, important point to get the mark, alpha cells stop releasing glucagon. There's no point releasing insulin if you've got the same amount of glucagon being produced as well. So this insulin binds to the liver and muscle cells, the specific receptors found on them. And then this will cause the removal of glucose. If there's a fall in glucose, alpha cells detect it, glucagon is released, beta cells stop producing insulin. Now this binds just the liver cells and more glucose is released. Now there are a few actual reactions that go on which you need to know about. Right. Insulin has certain effects. It will cause more glucose to enter cells through glucose channels. Glucose in the cell is converted to glycogen for storage, that's glycogenesis. More glucose is converted to fats, and more glucose is used in respiration. Overall, you're using more glucose and converting it into glycogen, which, as you remember from AS, is the storage molecule, so you haven't got as much actual sugar in your blood. If the blood drops too low, you have the conversion of glycogen to glucose. That's glycogenolysis. You use more fatty acids in respiration because, of course, you can not just use glucose in respiration, except brain cells. Brain cells have to use glucose in respiration. The production of glucose by conversion from amino acids and fats. So yet again, more glucose, and this is gluconeogenesis. And that's what occurs. Also, when you bite a liver cell, there are special liver cells called hepatocytes, and these are designed to do metabolic processes. Now, the control of insulin secretion is just a reaction you need to know about. So, here's a diagram. At number 1, the cell membranes of the beta cells contain both calcium ion channels and potassium ion channels. Remember, from the nerve section, you need to know about these ion channels. At part 2, the potassium ions channels are normally open, so potassium ions flow out. Yet again, you've got a potential difference across the membrane. Now, one side isn't negative, one side isn't purely positive, because they're just all plus ions, positive ions, but the relationship, it is negative compared to the outside. Now, yet again, a potential difference is about minus 70 millivolts, with the more positive on the outside, more negative on the inside. Now, at 3, when glucose concentrations outside the cell are high, glucose molecules diffuse into the cell, just diffusion. The glucose is quickly used in the metabolism to produce ATP. That's at 4. At 5, as you can see, ATP is being produced. At 5, the extra ATP causes potassium channels to close. At 6, the potassium ions can no longer diffuse out, and this alters the potential difference across the cell membrane. It becomes less negative inside, it depolarizes. The change in potential difference opens the calcium ion channels at 7. Calcium ions enter the cell and cause the secretion of insulin by making the vesicles containing insulin move to the cell surface membrane, releasing insulin by exocytosis. If you remember, that is similar to the synaptic knob. Calcium ions will bind to the vesicles, causing them to move to the outer of the cell and release whatever they have inside them, whether it be, in this case, insulin, or in nerves you had neurotransmitters. Diabetes, or diabetes mellitus. Now, this is a disease in which blood glucose concentration cannot be controlled effectively, and there are 2 types. In particular, you have hyperglycemia, is when you have blood glucose concentration which is too high. Think a good way of remembering it is if you are particularly hyper, you have a lot of sugar, hyperglycemia. And you have hypoglycemia, so you have hyper with ER, hypo with an O. Hypo is a state in which blood glucose concentration is too low. 2 types of diabetes, you have type 1 and type 2. Type 1 is insulin dependent. You have it from childhood. About 50% of type 1 diabetes is is diagnosed before your 20s. You get it very early on. What they think occurs, I'm not 100% sure, but they reckon the immune system attacks beta cells, killing them. This means you can no longer manufacture insulin. So there's no insulin to lower blood glucose levels. Type 2 is non-insulin dependent. We get it as we age due to a lot of other factors, such as obesity, if you have bad diet, family history, and also being Asian or Afro-Caribbean tends to mean you're more likely to have diabetes, generally because you are less used to having sugary food, so it has a bigger effect on you. Now, this is what happens here is the receptors decline on the liver and muscle cells. So you have the same amount of insulin, even though it can decline, but usually you have the same levels of insulin, but it's got nothing to bind onto, so you can't have an effect. And this is caused by environmental factors. Treating diabetes. Type 1 is usually insulin injections, or you eat lots of sugary food as well, you can. But you generally have insulin injections. Now, type 2 can be also controlled by the same thing, but it's usually a control of diet to make sure you're not eating as many sugary foods, and you can also have drugs and other things, but it's generally control of diet. Now, ways they think we can start curing it is genetically engineering insulin. Before, we were genetically engineering pig insulin. Now, this had lots of, bad moral objections of using animal insulin. But we can genetically engineer human insulin. Now, there are lots of advantages of this. It's an exact copy, so it's less likely to be rejected. It's going to be faster acting and more effective. You're less likely to develop a tolerance to insulin if it's your own insulin. There's low risk of infection. It's cheaper, and it's more adaptable to demand. So this is good, and people are less likely to object if you're using human insulin. We can also use stem cells to treat type 1 diabetes. So if you have more beta cells, which can be created using differentiated stem cells, you then can produce more insulin. Heart rate. Now, here is a diagram. It is very important to control heart rate. That is how fast everything's being pumped around your body. Now, heart tissue, as you remember, heart muscle is myogenic. It can initiate its own contractions because we have the sinoatrial node, which creates a wave of excitation, which goes through the AVN, which is the avioventricular node, and down the Purkinje tissue, which goes to the ventricles, causing them to contract from the base up. Now, the heart is supplied by nerves from the medulla, so another medulla oblongata of the brain. These nerves connect to the SAN. They do not initiate a contraction because the cells are myogenic, but it causes how frequently they occur. Now, there are certain things which affect the heart rate and what causes it to speed up and slow down. And there are two nerves you need to know about. There's the accelerator nerve, which will increase the frequency of heart rate, and the vagus or decelerator nerve, which decreases heart rate. Now, if you look here, there are a few things. High blood pressure detected by the carotid sinus. Now, this will cause a decrease, because if you've got a lot of high blood pressure, you want to slow it down. If you've got stretch receptors, muscles dead, if they're activated, then they will cause an increase. Low pH in the blood means that means acidic because of CO2 levels. If there's a lot of CO2, you want to get rid of that quicker, so you increase heart rate. I'm going to explain that a little bit more now. Movement. So that's what stretch receptors are. They detect movement. They send impulses to the cardiovascular centre, informing it that extra oxygen is needed. If you need more oxygen, you're going to increase the heart rate. More blood's pumped around, more oxygen gets around. It also means you'll probably increase your breathing rate. Now, pH, as I explained, we're going to get more carbon dioxide, and we're also going to get, through anaerobic respiration, lactic acid forming, which also lowers pH. Adrenaline. Now, adrenaline is secreted in response to stress, fight or flight response. Now, the presence of adrenaline in the blood increases the heart rate, getting the body ready to have that response of fight or flight if we need to do that. Blood pressure. Now, blood pressure again is monitored by stretch receptors in the walls of the carotid sinus. There's small swelling in the carotid artery. If blood pressure rises too high, in exercise, stretch receptors send signals to the cardiovascular system, and this reduces heart rate. And you also have CO2. Yet again, I explained that. And that is all. Conclusion. Hormones are chemical messengers. You have endocrine, which secrete hormones directly into the blood, no ducts, and exocrine, which secrete. You have actual chemicals, not hormones, going into ducts to specifically where they need to go. You have protein hormones and steroid hormones. The pancreas is an endocrine and an exocrine gland. You have the hormones you need to know about are adrenaline, insulin, and glucagon. Adrenaline gets the body ready for responses and activity. Insulin lowers blood glucose concentration. Glucagon raises blood glucose concentration. Diabetes occurs when you either cannot produce insulin, type 1, or you have no receptors for the insulin to bind to, type 2. And you also need to be able to control heart rate using hormones. So thank you for watching. As usual, any questions, leave in the comments or email me, like, comment, rate, and thank you for watching. Goodbye.